Hey guys, welcome to the first video of this series, which is going to be biochemistry for the MCAT. These are taken directly from the Kaplan MCAT books. You can buy any edition, really, it doesn't matter. I think from like 2017 Kaplan up. I had the latest edition, so I was reading it on my Kindle. So basically what my point is for these videos is that you want to make sure that you read the chapter, the corresponding section that I'm going to cover first, kind of like before a lecture, and then come here and then just get like a review, make sure you know all the concepts, make sure you all know all the key things. For these videos, I also expect you to have some science background, like no gen chem, biology, things like that, because these Kaplan books do give like an overview. They don't give like an in-depth, detailed explanation of little concepts. So today we're going to be talking about enzymes as biological catalysts from chapter 2. So enzymes are biological catalysts. What this means is that it's a catalyst. It increases the rate of a reaction. So catalysts help the reaction proceed at a much faster rate. And that's really what you have to keep in mind. Enzymes affect kinetics. They affect the rate of a reaction. What enzymes don't do is they don't affect the enthalpy of a reaction. They don't affect the thermodynamics of a reaction. They don't affect the equilibrium position of a reaction. And we're going to be mentioning this over and over again. Although they don't affect the equilibrium position of a reaction, they help the reaction get to equilibrium faster, but they don't affect the equilibrium state itself. So, and also enzymes don't change during the course of a reaction. They don't like degrade or anything. Enzymes are the same, which is why enzymes can be used for many substrates, because they can be reused and reused and reused. They're never used up because they don't change during the course of a reaction. So this is really important, this slide right here, because there's a lot of MCAT questions that come from these topics. They love like confusing kids. They love asking, do they know what it does? Do they know what it doesn't do? So just to review, enzymes lower the activation energy of reactions. They increase the rate of a reaction, so they alter the kinetics of a reaction. They do not alter the equilibrium constant. They are not changed, or consumed in the reaction. And something we'll talk about in the next sections to come is they're pH and temperature sensitive. So they have like an optimal pH and an optimal temperature at which they function at. And if you go above or below these kind of optimal ranges, the protein can become denatured, So, and they don't affect the overall Gibbs free energy of the reaction. They don't affect the change in G. And they're also specific for a particular reaction or class of reactions. We're going to be talking about the classes of enzymes today. So enzymes act on molecules called substrates. And something that's mentioned in the book is enzyme specificity, which means a given enzyme will only catalyze a single reaction or class of reactions with these substrates. So enzymes are specific, and they have specific reactions that they catalyze. And something I want you to note down really is that enzymes, although they have many different names, they all end in ACE. And that's the way you can remember it during the exam when you're reading these really dense passages. You can just look at the end of the name, and if it says ACE, you know it's an enzyme. And I picked some like really big sounding, proteins: beta-galactosidase, hyaluronide, I don't even know how to pronounce it, lactase. Anyways, all of them end in ACE, and you know that it's an enzyme. And you know the fundamentals of an enzyme, and that's overall what they're going to ask you. They're going to ask you the basics, the fundamentals. So these are the different classes of enzymes. Instead of writing it out, I just wanted to have like a diagram. And this diagram is from Jack Weston, so shout out to him and to that website. But we have oxidative reductases at first. What oxidative reductase enzymes do is they transfer electrons, and it results in a change in the oxidation state. So an example would be a dehydrogenase. Something I wanted to talk about these two arrows that are leading here and here is that in these redox reactions, the reductant is the electron donor and the oxidant is the electron acceptor. Something else I wanted to mention was Leo goes Gur. So that's how I remembered where reducing and oxidizing, what they mean is Leo, loss of electrons, oxidizing, Ger, gain of electrons, reducing. So in these cases, the reductant becomes oxidized and the oxidant becomes reduced. Because if you, it's kind of reductant and oxidant are the same thing as reducing agent and oxidizing agent. So you can see it either way. You can see reductant, oxidant, or reducing agent and oxidizing agent. And just keeping those in mind, that the reductant is the electron donor and the oxidant is the electron acceptor, and also keeping in mind Leo goes Ger and how these work together will be really, really useful for you. Because they can ask it either way. They can ask, does the reducing agent become oxidized? Does the oxidizing agent become reduced, etc. Second, these deoxidoreductases, as I said, take part in oxidation reduction reactions, so redox reactions, and they often have a cofactor because there's a transfer of electrons, so they need a cofactor like NADP plus or NAD plus to basically carry these electrons. They're electron carriers. They carry these electrons and help assist in this transfer of electrons. Now, transferases right here, they transfer functional groups from one molecule to another. So kinases, for example, would be transferring a phosphate. Hydrolases, they break down a covalent bond using water. It would be phosphatase, protease. So phosphatases, they break down phosphate bonds. They remove them from compounds using water. Lyases break down covalent bonds without water or oxidation. So an example would be a decarboxylase. And keeping this in mind that a hydrolase uses water and a lyase does not use water in the breakdown of a covalent bond is important. Now, isomerases rearrange bonds within a molecule. So if you see like a mutase or something like that, you know it's an isomerase. And ligases, they form covalent bonds between two large molecules. And having an example is unimportant. So ligases you're probably going to see in the context of DNA replication as we continue on. So something that was mentioned in the Kaplan book is that lyases, on top of the fact that they don't use water in breaking covalent bonds, they usually act on smaller molecules. And something else I want to mention is that these enzymes that we mentioned, they can also perform the reversible reaction. They can perform the reaction in the backwards direction. So obviously in the forwards direction, lyase would be breaking a covalent bond, but a lyase can also function as a synthase, right? To make a covalent bond, to construct it instead of break it. So synthase is the opposite of a lyase, and that was something that was mentioned in the book. I'm not 100% sure if that would be important on your exam, but something to keep in mind. Second, lyases act on smaller molecules and ligases act on larger molecules. So this is in the context of obviously lyases break down, synthase build up, ligase build up. But lyase and synthase, small molecules, ligase, larger molecules. So I want you to recall that thermodynamics relates the relative energy states of a reaction in terms of its products and reactants. And two concepts that I want you to really get down is an endergonic reaction and an exergonic reaction. So an endergonic reaction requires energy input. What this means is endergonic reactions, the delta G, the Gibbs free energy, is greater than 0. So it requires an energy input. It is not spontaneous. For exergonic reactions, energy is given off, meaning that the delta G, the Gibbs free energy, is less than 0. So energy is given off this reaction. This exergonic reactions are spontaneous. And the way you can really remember this is endo stands for in, so taking in energy, non-spontaneous. And exo stands for out, releasing energy, which would be spontaneous. So this is just a picture of showing us what an exergonic reaction looks like, also how an enzyme affects the activation energy of a reaction. So we can see that this is the transition state without the enzyme. This is the transition state with the enzyme. We can see that the overall energy change does not change, right? We added the enzyme, but the change in G does not change. Also, the change in H does not change either. So remember, we said that enzymes do not change the thermodynamics of a reaction. They only make the reaction go faster. They change the kinetics of a reaction. They also do not change the equilibrium state of a reaction. They only make it faster to get to the equilibrium state. So the overall reaction, change in G, is not affected. We have this transition state, the highest energy, right? We have free energy on our y-axis. We have reaction progress. So our transition state is our highest free energy. Then it goes down, exergonic. So in exergonic, our final state is lower energy than our beginning state in free energy. And then when we add a catalyst, or look at our activation state, look at our activation transition state, it goes all the way down. Our activation energy decreases. And that's what enzymes do. That's how they make it go faster. If you have a higher hill to climb, well, you're going to be tired. It's going to be so much harder for you to get up this. But if you have a smaller hill, if you have something that's more manageable, it makes it go faster. That much, you'll get over the hill that much faster, right? So that's how I want you to remember it. And here, activation energy, enzymes decrease it. Change in G and change in H, enzymes do not change it. And something you can also put in here is enzymes do not change the equilibrium constant. They only make it faster, reactions faster to get to the equilibrium state. And this would be an exergonic, negative change in D, a spontaneous reaction right here. And I just found this on YouTube from Bio Ninja. I thought this would kind of drive the point home, is that exergonic reactions without an enzyme, activation energy is much higher. With an enzyme right here, the activation energy is lower right there. And the activation energy, let me tell you exactly what the definition is the change in energy from the transition state to the energy of the reactants. So this would be the activation energy. And let me pick up a different kind of color. So for this would be the activation energy right there. And for the other one, it would go up here from the reactants. So it's the difference in energy from the transition state to the energy of the reactants, the free energy of the reactants. And vice versa, if it's right here, then it would be the difference between this and this. So that's very important to keep in mind, because that could be a question that they could ask you. So here we have, with enzyme, our transition state is lower in energy, makes the reaction go that much faster, less of a hill to climb. Remember, change in G does not change. And then endergonic, I wanted to show you what an endergonic reaction looks like. It's like a upward slope. Instead of ending with a lower energy, we actually end with a higher energy. And that makes sense, right? Because endergonic means we take in energy. So we should be ending with a higher energy than we started off with. So then with no enzyme, our transition state is super high. We have a really steep hill to climb. With an enzyme, our transition state is less intimidating for us to climb over. Remember, change in D does not change. And just to repeat the same concepts to really bring it home. So enzymes, they do not alter change in G and change in H, just as a review. And also they do not alter the equilibrium constant. They do not alter the equilibrium state. They only make it faster to get to the equilibrium state. They do not alter the equilibrium state, as I just said. They do not change during the reaction. They stay the same. They don't get degraded. They don't get changed. They stay the same. And that helps because that means one enzyme can act on so many different substrates, So we need less enzymes and more substrates to do the same reaction. So enzymes ensure the equilibrium state is achieved faster, but they don't change the equilibrium state itself. And enzymes affect the rate, the kinetics at which a reaction occurs by lowering the activation energy. So thank you guys for watching. This is the first episode of this series. It's section 2.1. Tomorrow I will upload section 2.2 for you to watch. Thank you. Bye.